The immune system is composed of three main components: organs and tissues, some specialized cells, and a wide variety of molecules. These three components are highly interconnected and work in a coordinated manner to protect us from external threats, such as the countless pathogenic microbes and the toxic agents that can enter the body from the outside, and also protect us from internal threats, such as our own diseased, damaged, distressed, or dying cells that may arise from within the body and may potentially cause harm or even transform and become cancer. Now let's start with the first of these components, which is the organs and tissues. This component is composed of the lymphoid organs and the lymphoid tissues associated with the mucosal epithelium. The lymphoid organs and tissues are characterized by being full of a very large number of lymphocytes. And these lymphoid structures are distinguished according to their function and are categorized into either primary lymphoid organs or secondary lymphoid organs. The thymus and the red bone marrow are the central or primary lymphoid organs. And these two organs is where generation, maturation, and differentiation of your lymphocytes takes place. B-cell maturation takes place at the bone marrow, while T-cell maturation takes place at the thymus. Now the rest of the lymphoid structures are considered peripheral or secondary lymphoid organs, and they include the lymph nodes, the spleen, and the mucosa-associated lymphoid tissues. The secondary lymphoid organs is where most lymphocyte activation and proliferation takes place. So after lymphocytes are generated in the primary lymphoid organs, they will go to the secondary lymphoid organs where they get activated. Now we'll have a couple of words on each one of these organs to learn their main role in the immune system. Let's start with the primary lymphoid organs, beginning with the red bone marrow. The bone marrow is the site of hematopoiesis that give rise to all the formed elements of blood, including the leukocytes, which play an essential role in the immune system. The bone marrow is the site of both B and T cell origin, but when it comes to the development and maturation of these lymphocytes, B cells remain in the bone marrow for the majority of their maturation, whereas the progenitors of T cells will have to exit the bone marrow and travel to the thymus to complete their development. The thymus is also a primary lymphoid organ, and it is the site of terminal T cell maturation and differentiation. And it is where T cells receive their education to finally become helper and cytotoxic T cells. Now part of this education is programming these lymphocytes to recognize self and not react against it. And that is called self-tolerance. This process is very essential for normal immune function. And without it, our lymphocytes will react against our own tissues and cells, resulting in autoimmunity. So these two are your primary lymphoid organs, and once again they are called primary because the main process of generation, maturation, and differentiation of your lymphocytes takes place there. Next, we will move to the secondary lymphoid organs, which are the lymph nodes, the spleen, and the mucosa-associated lymphoid tissue. These secondary lymphoid organs function mainly as areas that trap or filter foreign antigens. The lymph nodes are designed to filter pathogens incoming from tissues. So antigens that are filtered from your tissue spaces will be carried in lymph and will travel via multiple afferent lymphatic vessels towards your lymph nodes. The spleen, on the other hand, is specialized in filtering pathogens incoming from blood. So antigens that are filtered from your circulation will be carried in blood and will travel via blood vessels towards your spleen. Now, the third of these secondary lymphoid organs is not actually an organ, but rather a disseminated system of small collections of lymphoid tissues. And this collection of lymphoid tissue is referred to as the mucosa-associated lymphoid tissue. The mucosa-associated lymphoid tissue is all over the mucosal surfaces of your body and is subdivided to many components according to its location. For example, the lymphoid tissue in the gut is called the gut-associated lymphoid tissue, and it includes the Peyer's patches, the appendix, and some other diffusely scattered lymphoid aggregates and lymphoid cells throughout the gastrointestinal tract. Another example is the nose or nasopharynx-associated lymphoid tissue, which consists of our tonsils that form together a lymphatic ring called the Waldear's tonsillar ring. The mucosa-associated lymphoid tissues in general have one thing in common, which is being responsible for filtering antigens encountered along the mucosa. So these secondary lymphoid organs altogether filter our tissues and body fluids and receive the filtered antigens. And by doing that, they provide sites for lymphocytes to interact with those antigens in a process called antigen presentation. So the secondary lymphoid organs are like a bridge where our innate antigen presenting cells interact with our adaptive T lymphocytes. And once this interaction occurs, these lymphocytes will get activated. And then will start proliferation and increase in number in a process called clonal expansion, before their differentiation into effector cells and also antibodies, both of which are specific to that particular antigen. These effector cells and antibodies are capable of mounting an adaptive immune response that eradicates the pathogen. Now let's move to the second component of the immune system, which is the immune cells. The immune cells are distributed throughout the body. They could be circulating in the blood, or traveling in the lymphatic fluid, or could be tissue-resident immune cells that reside and form an integral part of various epithelia. The immune cells are born in the bone marrow in a process called hematopoiesis. They develop from bone marrow's multipotent stem cells that later differentiate and give rise to two major cell lineages of progenitor cells, the myeloid progenitor and the lymphoid progenitor. These two stem cells will eventually become the different types of white blood cells. The common myeloid progenitor gives rise to other progenitors, and we will not go in details for all those progenitors for now, but just keep in mind that the myeloid lineage will give rise to neutrophils, eosinophils, basophils, mast cells, monocytes that give rise to macrophages, and some dendritic cell populations. Dendritic cells actually can arise from both myeloid and lymphoid cell lineages. The common lymphoid progenitor also gives rise to many progenitors that will end up in forming the following cells: the innate lymphoid cells, and an example of such cells is the natural killer cells. The T-cell progenitors that differentiates into many subtypes, such as helper, cytotoxic, and memory T-cells. The B-cell progenitors that differentiates into many other cell types, but most importantly into plasma cells that secrete antibodies. Now, as an introduction, you should know the answer to the following questions. Which of these cells are phagocytic? Which cells are part of the innate and which are part of the adaptive immune system? And what cells connect these two systems together? Finally, you should know which of these cells are antigen-presenting cells. And the answer to these questions is pretty straightforward. All the cells of the myeloid lineage are phagocytic, meaning they can engulf and ingest foreign particles, but their phagocytic capacity varies. The two major phagocytic cells that eat up the most are the macrophages and the neutrophils, and they are among the first cells to encounter and respond to pathogens. Now, all the cells of the myeloid lineage are part of the innate immune system, and all the cells of the lymphoid lineage are part of the adaptive immune system, with the exception of the innate lymphoid cells, which are part of the innate immune system as their name implies. And natural killer cells actually are a subset of those innate lymphoid cells. Now, what connects the innate and the adaptive immune systems together is the process of antigen presentation that we mentioned earlier, and which takes place in the secondary lymphoid organs. And our main specialized antigen presenting cell is the dendritic cell, which can grab and capture foreign antigens. These antigens will then get internalized into the cell and will get processed and prepared, and then will be carried and displayed on the cell's surface in preparation for the process of antigen presentation, where this antigen will be presented to a T cell, and this is what starts an adaptive immune response. So the antigen presenting cells are mainly the dendritic cells, but in certain situations, macrophages, monocytes, and even B cells can also act as antigen presenting cells. And the take-home message here is that those antigen-presenting cells form a crucial link between the innate and the adaptive arms of the immune system by presenting antigens to lymphocytes. And this process takes place at the secondary lymphoid organs, which are like a bridge that provide an area for both arms of immunity to interact. We talked about the organs and tissues and also the cells. Now we will move to the third component, which is the protein molecules. These molecules are responsible for mediating the communication within the immune system and are also responsible for regulating the immune response. And it is due to these molecules that your immune cells are able to move and interrogate their environment for evidence of anything foreign or abnormal. One very important example on such protein molecules are the antimicrobial proteins and peptides that are found in various body fluids. These antimicrobial proteins are extremely important in barrier protection, and they provide defense against pathogens by inhibiting their growth or by directly killing them. Another example on a set of molecules that are essential for innate immunity are the pattern recognition receptors. These receptor molecules act as sensors, giving our cells the ability to discriminate between self and non-self, and they provide an initial recognition of invaders and a wide range of danger signals. So it is due to these receptors that our immune cells are able to recognize certain patterns and shared sequences of many pathogens, and also recognize patterns associated with diseased, damaged, distressed, or dying cells. And once this recognition is achieved, what actually happens is that this recognition induces and triggers a variety of responses and defense mechanisms in our immune cells against the dangers that they encounter. And an example of such response that result due to the interaction with pathogens is the secretion of other protein molecules called cytokines and chemokines. These cytokines and chemokines convey important signals that can initiate a local inflammation and can propagate the immune response by recruiting lots of cells, molecules, and fluid to the site of infection. And these protein molecules is what makes your white blood cells move and attract them along their concentration gradient and allow their migration directly towards the heart of infection or inflammation. And it is due to these protein molecules which are pro-inflammatory that we see the systemic effects of fever and leukocytosis during the acute inflammatory response. Some of these cytokines also support precursor cell growth and the differentiation and activation of various immune cells. Another example on a set of protein molecules is the complement proteins. Complement proteins enhances our defenses against pathogens mainly by promoting inflammation, enhancing phagocytosis, and lysing microbial membranes. One example is the complement protein that is called C3b, which is an opsonin that enhances the process of phagocytosis. C3b attaches to the pathogens and basically acts as gravy to make the pathogen more delicious and help the macrophages eat or phagocytose more readily. Other enzymes and protein molecules are involved in breaking down the ingested material. And that's essential not only for pathogen killing, but also is crucial for initiating the adaptive immune response by the process of antigen presentation. This process also needs other protein molecules. For example, we have what is called the major histocompatibility complex class II molecules. And what these molecules actually do is that they grab parts of that ingested and degraded pathogen and takes it all the way to the surface of the cell and displays it in order to present it to T cells. Other proteins act as glasses for your lymphocytes, and without them they can't see those parts of the pathogen that is being presented. And an example is the T cell receptor, which is able to recognize peptides found on the surface of specialized antigen-presenting cells. Some protein molecules are like barcodes on the cells, and scanning them by immunocytochemical methods help us differentiate between various populations of leukocytes that even our best microscopes can't distinguish. And such molecules are called cluster of differentiation molecules, and they are commonly used as cell markers. So we have lots of protein molecules acting on our immune system, and they are so numerous. We have lots of cytokines, receptors, antibodies, complements, cell adhesion molecules, and cluster of differentiation markers. And the take-home message about these protein molecules is that they are essential for mediating communication within the immune system. They regulate many functions. They influence and control cells' behavior. They sense danger and recognize intruders. They stimulate and activate things and inhibit others. They are like the nervous and endocrine systems of our immune system, and without them our immune cells would be paralyzed. So these three components that we just discussed is what forms our immune system and what makes it work. Now, for the ease of discussion and to better understand immunology, we'll put the immune system into two separate responses and divide it into two arms: innate immunity and adaptive immunity. Although in reality the fact is that our immune system is highly integrated and these two arms work together closely, and some of their components actually can't be separated. But at the end, these two systems collaborate to give our body the best possible defense against disease. Innate immunity consists of barriers, phagocytic cells, and a limited number of pattern recognition molecules that are inborn and encoded by genes directly inherited from an individual's parents. Innate responses are limited in specificity and are unable to distinguish between small differences in foreign antigens, but they're actually extremely fast and start working immediately after contact with pathogens, and are aimed at preventing infection or quickly eliminating common invaders. And by doing so, innate responses buy time until our adaptive responses gets into action. Adaptive immunity, on the other hand, consists of T and B cells and their highly diverse antigen-specific receptors that are not inherited but rather generated randomly by gene rearrangement. Adaptive immunity is slow and takes days to form because it relies on expansion of an army of effector T cells and antibody-producing B cells. One particularly significant and unique feature of adaptive immunity is its ability to learn from its encounters with foreign antigen and also store this information for future use. And that's the immunological memory that gives our immune system the ability to respond much more swiftly and with greater efficiency during a second exposure to the same pathogen. We came to the end of this introduction. Now if you want to get the best of what we're doing in ACE Immunology, check out our lecture notes and also test your learning through doing multiple choice questions. And don't forget to subscribe to our channel. Thanks for watching and see you in the next video.